Hi, I'm your instructor, Dr. Hankey. I'd like to go over the week 5 discussion questions right now, particularly question number 2. You all did really well with number 1. You would not take an antibiotic for a viral infection because antibiotics are designed to attack specific bacterial components and structures. Viruses don't have those, so they're ineffective, and they can actually weaken us by eliminating the good bacteria that we have on our bodies. So week 5 discussion question number 2 talked about describing the replication process for one of the following types of viruses, either a single-stranded DNA virus or an RNA retrovirus. We could do this with any type of virus, single-stranded DNA, double-stranded DNA, a single-strand RNA with the reading strand or with a negative strand of RNA, or a retrovirus, which is lysogenic. The question asked you to describe the steps of infection. That would essentially be the same for all, which is attachment. You have to have the entry and the decoding, or uncoding rather, and then the components need to be synthesized, meaning any part that goes into the cell, the infected cell, has to be manufactured so it can be put into new viral particles, virions, one complete viral particle, to exit the host. And then maturation is when all those separate parts, the capsomeres that will go together to make the capsid, those protein molecules, and whatever nucleic acid went into was in the original virus. And so I'd like to just go over this just to give you an idea. This lets you play with what you learned about in genetics in chapter 8. Humans, animals, were pretty straightforward. We copy our DNA, the entire genome, in order to make new cells for replication, for growth, or for repair. Or to increase population size of single-cell organisms. And from there, we take that DNA and we go through transcription to make messenger RNA, or all the types of RNA, to then translate those into proteins. And primarily, the important ones are our enzymes. Those are our busy bees, our builders that are going to make all the components that will then make us, or a new functioning cell. So bacteria play around with that a little bit. That bonus question on your exam where you took different nucleic acids and could go forward or backward because of the complementarity, because of complementary base pairs, whatever strand you have in the process, you know and are able to determine what the other strand will look like. Any molecule can be used as a template for its corresponding complementary base pair molecule. We only go in one direction, transcription to translation. Viruses can go in different directions. They have the enzymes that let them do that. So we have retroviruses. So let's take a look. First, we'll start with a double-stranded DNA because it's what we're most familiar with, because we have double-stranded DNA. So whatever the virus carries into the cell when it infects it has to be the same thing that comes out in the newly synthesized virions. So when the virus is lytic, when it lyses the new cell to spread to neighboring cells, those new virion particles that spread have to be identical to the virion that came in. In double-stranded DNA, the virus enters with a double strand of DNA and the proteins that make up its capsid. So we enter the host cell, and those are the parts that we have to replicate. So we're going to use those complementary base pairs to help us. And this is really something that can snowball quickly because we use whatever molecule we have to make more of the molecules we need. So once we enter the host cell, what goes in, our double strand of DNA, also has to come out. We'll use semi-conservative replication, the same process all our cells use to make copies, to make more double-stranded DNA that will then be packaged into the new virion. So we come in with a double-stranded DNA. We make copies of both the positive and negative strands, using each strand as a template to make more. And we quickly make lots and lots of new double-stranded DNA. We also use our double-stranded DNA to go through transcription to get messenger RNA that can be translated into the proteins that will make up the capsid or the capsomere. During synthesis, this is what's going on. We're making the parts. So when we say synthesis, here's our synthesis, this portion. We're making the new parts that have to leave the cell. Maturation is when all those pieces come together to make a new virion, a nucleic acid, in this case double-stranded DNA, that's enclosed within those protein capsomeres that making up the capsid, the protein coat. So that's the process that a double-stranded DNA molecule would go through. For a single-stranded DNA, what the virus carries into the cell has to be the same as what the newly synthesized virions carry out. So in this case, we come in with a single strand of DNA, our reading strand. This is what enters the host cell. It also is surrounded by that protein coat. So what comes out has to be more of those proteins that build the protein coat, as well as that single-stranded DNA. So what we're going to do is use DNA polymerase to take our positive DNA strand, use it as a template to build negative DNA strands, which gives us this intermediate double strand. We then use the positive to build more negatives so we can use the negative strand to make more positive strands. So you can see how this snowballs. The more negative strands we have, the more positive strands we can build from that template. So we'll go back and forth, making more negatives to make more positives with this double-stranded intermediate, so that we can generate more positives. We'll also be generating more negatives, but those are just going to be garbage strands that will be eliminated from the body as garbage, along with the cellular debris after the cell lyses when the virions break out of the host cell. We'll also use those newly created double strands of DNA as templates to make RNA, which the RNA will be translated into those viral proteins. So we'll have our viral proteins, the capsomeres that will go in to make the capsids that will surround the single strand of DNA at maturation, and our newly formed virions will go out. All of the RNA and all of the negative DNA will just be eliminated as waste products after that cell lyses. And then finally, our retroviruses. What's coming in with a retrovirus is a single strand of RNA, but because these are lysogenic, they're going to go into a latent phase where they will incorporate their DNA into the host DNA. But they don't have DNA coming in. What they do have is this enzyme, a reverse transcriptase, which, just like the name says, it transcribes in reverse. Transcription is typically DNA used as a template to make RNA. In this case, we're going to take our RNA, and because of complementary base pairing, we can use it as a template to make DNA. Reverse transcriptase is also known as RNA-dependent DNA polymerase. This molecule builds DNA from RNA. So we're going to take our single strand of RNA, and we are going to build a single strand of DNA from it. It's the template, so it's the complementary base pairing. We'll use that as our template to make the positive strand so that we have our intermediate double-stranded DNA. This matches our host cell's DNA. This is what gets incorporated. At this point, the virus can go into the latent portion of its lysogenic cycle, incorporate into the host DNA, and it may or may not ever be expressed. Many people carry the shingles virus, but it's never expressed, or the herpes virus, but it's never expressed. It can stay latent for the lifetime of the host. Or it could come out of the lysogenic latent portion of that cycle and go into the lytic cycle. At that point, that DNA, that particular gene, those genes, that portion, would be transcribed back into the viral mRNA, and we can use both portions to copy positive and negative RNA, both strands of the RNA, because one strand can then be used as a template to make more of the other, meaning that we can make more of the proteins that we need during translation because we have more and more of those molecules. So that snowball effect again. We'll translate that RNA into both capsid proteins and the enzyme that we need. This virus has to carry its enzyme with it because host cells don't have this enzyme. We don't have the genes, the information for how to build it. So the virus has to carry that with it. So what then is going to leave the virus, we're going to make RNA from our DNA strands. Because we came in with a single strand of RNA, we have to leave with a single strand of RNA. We have to leave with all our viral proteins that came in. Those were both the protein coat and our enzyme reverse transcriptase. So synthesis is all of this, making these parts that we need. And maturation, they come together and they leave the cell. They lyse out of the cell in the lytic portion of the cycle. And again, with these, we could just go into this first step of becoming latent, incorporating the DNA into the host cell that is there forever, whether or not the virus ever enters this lytic portion. So I hope that helps. You can see why that bonus question, we did that so you can better understand how that complementary base pairing becomes critical for all the subsequent steps in the process. So hope that helps.